Okay, this is a sarcomere. The sarcomere is the smallest contractile unit of a muscle. It's going to be a part of the structure of a myofibril. It's made of both actin and myosin, right? And so the actin filaments, these small filaments, are going to be in a 6-to-1 ratio. There would be 6 actin filaments surrounding every one of the thick myosin filaments. The myosin filaments are going to be the larger of the two, and the myosin heads, these, are going to, when the binding site opens on the troponin complex, be able to bind from the thick filament to thin, and in a ratchet type manner perform the power stroke and shorten this overall sarcomere's length, like so, bringing it inward and contracting the muscle either completely or partially. This would be the Z disc right here, would be the Z disc or Z line, right? And so a sarcomere consists of from Z line to Z line, and that structure would be would have attached to the Z line or Z disc, the protein disc, a group of actin filaments. The actin filament itself is made of the yellow tropomyosin protein, and it's going to actually be wrapped around 2 actin filaments, fibrous proteins, and on each of those in the binding site where the myosin head is going to grab, you have what's referred to as the troponin complex. It's the release of calcium from the T2 or the terminal cisternae and the sarcoplasmic reticulum, and the binding of that calcium to the troponin complex that causes a conformational change in embedding of this troponin complex into the actin filaments, opening up the binding site so that these two things can interact and the sliding filament mechanism can ensue. The shortening of the sarcomere overall would be seen in the structure when the actin filaments, these structures here, are pulled inward into the H zone, which is this zone here. So this would be a relaxed muscle, and this relaxed muscle would only be said to be contracted if the actin filaments would be pulled into that H zone towards this M line at the center. The entire sarcomere is made up of an A band, which would be both actin and myosin here in the center of this structure. Then the I band would be the lateral edges, where just actin and the Z disc are located. At the center, as I've already said, is the H zone, and in the middle of that H zone is the M line, where the actin filaments are pulled towards the middle of it. That's the sarcomere. All right, so each of those structures, each of the structures that I just discussed are found in this model here. And so this model is a muscle fiber. Right, a muscle fiber is going to be made up of myofibrils, each of these circular units, and if I were to bundle many of these fibers together, I would have a fascicle, and we'll talk about that in a moment. These myofibrils, these bundles here, are made up of actin and myosin proteins in a 6-to-1 ratio, and it's the arrangement of those proteins that give you this striated appearance that you see here on the model, the striations that are visible. Now, from the surface of this muscle, as you can see here, you have openings that go into the muscle through the sarcolemma. This is the phospholipid bilayer of the muscle, and so this particular opening right here, each of these openings lead into these structures, T-tubules, and that's how sodium gets deep into the muscle to allow for a complete muscle fiber contraction. Okay, so as this muscle, or this neuromuscular junction here, here's a neuron right there connecting at the neuromuscular junction to the sarcolemma of this muscle. You would have excitation, coupling of these two things together, and you would have sodium influx into this muscle fiber. And that sodium influx, if it reaches threshold, as we discussed in class, will result in an all or none action potential being generated and shortening ultimately of the through sliding filament mechanism within this muscle. But on this particular model, what I would hope that you guys know would be the neuromuscular junction. That's these two things together. This is the neuron axon. It terminates in an axon terminal here, or terminal boutons, and that is the motor end plate or synaptic cleft on the sarcolemma of this particular muscle. So the sarcolemma or surface is where we have an interaction between a neuron and this particular muscle fiber. Each of these small organelles would be mitochondria producing oxygen so that this muscle could continue to function over and over again for prolonged periods of time. Notice that on the surface of this muscle you have nuclei This is a multinucleate skeletal muscle, and therefore the skeletal muscle would need the ability to produce proteins in abundance over and over as you tear the muscle and you build muscle mass with each time it is used. Here, deep in the muscle, you had these openings, and these openings were the T-tubules. These T-tubules lead into these blue channels. This is the T-tubule itself, and notice it goes all throughout the muscle. Any area where there is nothing and it's just surrounding myofibrils would be, of course, the sarcoplasm of that muscle fiber. The T-tubules then, the blue channels, are surrounded on either side by the sarcoplasmic reticulum and extensions of that SR, and the extensions are called terminal cisternae. So this triad right here would have calcium ions on either side of it, and in the actual T-tubule that's open to the outside, you'd have sodium influx and the ability to fully contract this particular muscle fiber. It takes anywhere from 4 to hundreds of neuron axon terminals to innervate one muscle fiber. If I bundle many of these fibers together into a unit, like I would see in the body, each of these units, each individual unit is too long. Each individual unit then is what we just looked at in the muscle fiber. And so I know that from this model because here I see a neuromuscular junction. I see multiple nuclei, right? And I see that this particular unit right here would be a muscle fiber like what we just looked at in the larger model a moment ago. So this muscle fiber, when bundled with other muscle fibers together, forms a fascicle. And so this model is, entire model is a fascicle, and when I combine many fascicles together, I form a muscle tendon, and that's what would be on the models of the arm and leg and torso models that we've been studying. The compartmentalization of these layers or compartments within the muscle would be via a connective tissue at either dense regular or dense irregular connective tissue, mostly dense irregular, unless it's the tendon itself and it's being reinforced in one direction. But this layering, for example, right here and on the individual fibers and on the muscle fiber that we just looked at, this layer that's white right here is referred to as the endomysium or paramysium of that muscle unit. And so if this is multiple fibers together, each individual fiber would be surrounded by endomysium. On the individual fiber model, this is endomysium. And then if I bundle all of those individual units together in a fascicle, paramysium surrounds that whole fascicle. And you can see what we showed first, the sarcomere structure seen here. There's the Z disc, there's another Z disc, this is the H zone in the middle, this is the A band here, and then on either side the lighter areas would be the I bands. And as I pull the I band inward and bring the actin filaments into the H zone toward the M line, I would be contracting these contractile units.